Today we're starting respiratory and we'll be doing anatomy, some anatomy lab. We'll continue that on Tuesday. Tuesday's another goggles, gloves, closed shoes lab. So even if it's warm, I don't know what it will be next week. But there's a sheep pluck dissection. I wish we had sheep pluck for everybody. I've done one lab with that and it was a lot more fun. But this will be a demo that you can get involved in. A sheep pluck is kind of like we have, oh you can barely see it, so the very last cabinet. It's the lungs, heart, all in one piece. A little bit of diaphragm, usually liver and gallbladder are intact with that as well. Our interest, and trachea, our interest is of course the trachea and the lungs. And we'll be placing a glass tubing into the trachea, to which is attached ventilator tubing and an ambu bag. The ambu bag is courtesy of my protest. About 12 years ago, we used to blow this up, put our mouths on the sterile glass tube and blow into the sheep lung. And they have recoil, elasticity and recoil. Yes. And I had to get my mouth far enough off of the glass tube in time, and the recoil shot and you get some blood into my mouth. And I said, I'm never doing this again. So we now extend it with the ambu bag and so on. When I did this at Sac State, each group of 4 had their own, and they had a table attached to, we have gas attached to these things, but it had air. And so instead of blowing into the tubing or pumping it, they would use the air from the air source tanks to expand it. So that's an observation. You can get involved with the pumping part, and with gloves you can palpate the lungs. After that, it's totally up to you. Usually there are some students that want to dissect into that, follow the trachea out to bronchi and so on, and observe or look at the heart connections with the bronchi and pulmonary arteries and veins. So that part is optional afterwards. But bring gloves if you want to handle it, and goggles in case there's more spraying of spit. There shouldn't be, but also opening up the larynx. But it doesn't come with the larynx, so we can't make the sheep talk, but you can open up the trachea and expand those components. I think everybody's here. I have a proposal to make to you. Well, we will have lab today, but if you'll allow me to make it a little bit shorter and talk a little bit longer, I have a memorial service to go to tomorrow, and it would be letting you out early and maybe getting back later, not at all for the other one. So I don't want to get behind already, but I think we can cover that material. So your patience, if I'll give you breaks and stuff for lab time as we go through, okay? And then we won't have class tomorrow. So you can come to open lab and utilize that time. And then on Tuesday, we'll kind of pull the lab together as well with the anatomy component, okay? I do have, as you've seen, the grades are posted for the first time ever. I got everything done before I left campus. And so I still need to photocopy the answers onto your labs. I'll do that during our first break, but I have everything to head back, okay? All right, so what we're looking for anatomy-wise is the air passageways. So removing air from the external environment, what we call the atmosphere, deep down into the lungs, and then back out again. So all the structures that the air molecules are going to pass through to enter deep into the lungs and pass out as part of the respiratory system. When we do physiology, we will follow the gas molecules across the respiratory membrane and into the blood, and what happens is it does the hemoglobin. So we'll be looking next week at air pressures and atmospheres and so on. So there are 3 components to respiration. The 4th one has been added here, but typically that's within the cell itself. That's the mitochondrial activity with oxygen that we covered last semester when we looked at cell organelles. And so the 3 parts of respiration, and we'll look at this in lecture next week, but we're going to cover the anatomical components, are pulmonary ventilation. Okay. And pulmonary ventilation is comprised of the movement of air into and out of the lungs. okay? And that's really inspiration and expiration. So it says external respiration, but it's inspiration and expiration. What we think of as breathing. And then the next part is external. So this is not the same as inspiration and expiration. Okay? External respiration is gas exchange. Both carbon dioxide and oxygen across the respiratory membrane. The respiratory membrane is a sandwich. Okay? So this is essentially the two epithelia, the air sac and the capillary. I use a different definition for air sac than the textbook does, so let me change this to alveolus and capillary. So going from the air to the blood, okay, is external respiration. And then internal respiration is gas exchange from the capillary to tissues or back. So we have the capillary and extracellular fluid. So again, this can be both directions, depending on whether we're talking about oxygen or carbon dioxide. So this would be carbon dioxide, and this would be oxygen. And the same on this side, carbon dioxide and oxygen. So today, we're going to pay attention to the structures of pulmonary ventilation. What move, not what, but the structures, the pathways that air moves into and across deep down into the alveoli. We haven't done embryology for a while, and there aren't any structures different for the fetus here than what we looked at before. But remember when the embryo closed and created a longitudinal tube that was lined with the endoderm that became the mouth and gut structure? So that is going to be this yellow structure right here, all right, the embryonic gut. And then we have a mouth and esophagus, and a bud grows out of the region that is going to become the esophagus. And so from here on out, these two structures are very closely related. You can see that when you look at our heart model. You probably did pay attention to the trachea and esophagus on those posterior aspects. So that's going to become the trachea. And after it grows, two buds occur. And each of those would be the primary bronchi. And then a continuous budding occurs as it grows out into the mesoderm of the thoracic cavity. And finally, we're going to see changes in epithelia because, I don't know if you recall from 430, but we had our greatest variety of epithelia in the respiratory system. Digestive tract just goes from non-cornified to simple stratified squamous to simple columnar. Respiratory tract is going to go from pseudostratified columnar to simple columnar to simple cuboidal to simple squamous, as we get deeper and deeper into the airways and need a thinner and thinner membrane. Okay? So that's the basic aspect of the embryology, is the origin, and then just continued budding and dividing afterwards. Now, when we look at these structures, they can be divided functionally and by location, right? By structure and by function. So here are the two zones that are described, and that's not clear completely what their function is, but once you understand respiratory it is. Conducting zone, there is no gas exchange, all right? This is merely a tube, or a series of tubes that air pressure, difference in pressure, pushes the air in or pushes the air out. So all of our epithelia there and layers of muscle and connective tissue are going to be too thick for gas exchange to occur. Once we get to the first section, the first airway that demonstrates a simple squamous epithelium at any part of it, we can have gas exchange. So the respiratory zone means gas exchange can occur. Maybe not across the entire wall, but in small portions of it. So the respiratory zone is a site, at least in part, of gas exchange. So any structure that has the ability for gas exchange to occur between across the simple squamous epithelia to a capillary. And there are isolated alveoli for that gas exchange to occur. We're in the respiratory zone. The next description is by location. So when you have an upper respiratory tract or a lower, well, this is still showing airways, so let me stick to this for a second. You don't have to worry about the transitory, but as you look here, we have it includes the nasal cavity, but lower respiratory zone would be upper respiratory tract and lower respiratory tract. So these structures are lower. This is cartilage. All right, and as we get down, notice in the respiratory zone, the first thing to appear are these little grape-like structures. These are alveoli, and they are formed of simple squamous epithelia. So gas exchange can occur at that point. Not here, but across the small alveoli, but at that level. And so that's why it's considered to be a respiratory zone. So structurally, we divide it into upper and lower respiratory zones. Not respiratory, sorry, upper and lower respiratory tract. So let me overlay these two against each other. And this is not a functional, so it's not the same break as we have with the zone. An upper respiratory infection would be a sore throat, okay, a cold. We're going to include the larynx in the upper respiratory tract. Sometimes we'll include it in the lower. We'll include it in the upper. And the laryngitis, okay, would be upper respiratory. So we'll have the nose, the nasal cavity. The nasopharynx. Remember, pharynx is another word for throat. And it has 3 regions. So we have our 3 regions of the throat, and then the larynx itself, or the voice box. So that's upper respiratory tract, and then a lower respiratory tract. So pneumonia, bronchitis, emphysema, any tracheal, bronchial, or lung disease is going to be a lower respiratory tract. So we'll start with the trachea. And as an overview, the trachea then divides into primary bronchi, secondary bronchi, tertiary bronchi. So these are just the level of those divides that we talked about with the embryology of it. Probably one more branch, but I'm not going to write that up there. And then notice the difference in the word, because this makes a difference in your spelling as you identify structures. We have bronchioles. So bronchioles are getting a smaller epithelium. They still have smooth muscle. They're not a site of gas exchange yet, but they do become that. So here's a bronchiole. Notice the difference. We'll go over these individually, but the bronchiole lacks the cartilage. And then when we first start adding an alveolus, it becomes a respiratory bronchiole. Right? So we're going to have bronchioles that we just identify as bronchioles. And then just before it becomes a respiratory bronchiole, it's known as a terminal bronchiole. But you have to be able to see the respiratory bronchiole to know it's going to become a highlighting in blue are now our respiratory zones. Remember the conducting and respiratory zones. So the respiratory bronchiole is the first of our respiratory zone. And now you guys in the back can't see it, so I'm going to move back up here to the top. And then we have alveolar ducts. And alveoli. Typically the sac are clusters of alveoli. So remember how we went through the lymphatic organs, unless you've dumped all that from your brain already. And we compared them by structure, which had capsules, which had efferent or afferent lymphatics, and so on. So as we go through, especially the lower respiratory tract, we'll look at the histology of that. And our first lab quiz will be just structure. And then we'll have a second lab quiz like we did with the endocrine system and reproductive, where there'll be the histology, the microscope slides, and so on. But we'll look at the histology of what's the epithelium. Is there cartilage present? Where's the smooth muscle? And so that's what you'll need to focus on is characteristics, both in lab identification and lecture identification, especially for the bronchi, trachea, and bronchi. All right, can I erase the board? You guys have those structures up there. So we're going to start here with the nasal cavity. Right? Where does air enter into the nasal cavity? So I'll leave that diagram up on the screen for a while as I attempt to draw it. I have a little trouble with lips usually, but we'll see how this goes. Through a pair of openings that you may know of as the nostrils. The openings, however, are not the nostrils. The nostrils are the structure around the opening. So the opening itself is called a nares, and this is known as the external nares, or nares, and you can go back and forth between single and plural. So we have two of those. Okay, if I were to stick my finger up my nostril, that would be the opening there. And then the space within the nostril is called the vestibule. So we're not yet in the nasal cavity proper. Okay? We passed through the external nares into the vestibule, and now we're in the nasal cavity proper. And remember the turbinate bones or the conchi? So those structures, we have the superior up quite high. That was difficult to see in the models because it was kind of up and ran into the middle and was kind of covered by that. So we have superior and middle and inferior nasal concha. Also known as turbinate bones. Now the first two belong to what bone? You guys remember from file 430, superior and middle?
Is that the sesamoid?
Which one? Not sphenoid, but it rhymes with it. Ethmoid. Those two are part of the ethmoid bone, and the inferior nasal concha is its own bone. Now in this diagram, obviously the perpendicular plate is absent, but that forms the nasal septum. And so, let's see, where should I put that over here? What two bones form the nasal septum? Remember when you look in the skull, you see a ridge right here, and the two bones are like this. So the bottom one is the vomer, and the top one is another process of the ethmoid. Yeah, the floor of the mouth is formed by the maxilla and palatine forming horizontal plates. The ethmoid forms a perpendicular plate. So the ethmoid provides the perpendicular plate. And again, the inferior structure is its own bone, the bone. But we can't see that because we have, this guy doesn't have much of his brain. Nose, and then there's no more brain up there. So forming the floor of the nasal cavity, we have two bones. So we have maxillary and horizontal plate of the palatin.
I'm getting hints.
I know it's back there somewhere. All right, so air flows around, under, and over the turbinate bones. And they have, in that mucous membrane that covers them, and you can see them on their model here, and the mucous membrane that covers them are large venous sinuses, which are wider than normal veins, right along the surface here, and close to the surface. So they provide both warmth and adding moisture to the air. Because as cold enters your lungs, it causes the smooth muscles of your airway to constrict, and we don't want to have a more narrow airway. We've got another structure here. Hair follicles. He needs to trim his hair, but that helps to filter out dust. So if you've been, riding on a car or a motorcycle and down a dusty road, you get all this grime crusted around your nose, and that is down your nose, and instead of going down into your airway. So now we're ready to leave the nasal cavity, and we pass through another pair of openings before we actually enter the nasopharynx, and I probably have mine. So this is sometimes difficult for students to recognize by the fact that we have one opening there and there's really a pair. So typically it becomes clearer if we take, if this will work. I already have this mandible removed, so let me take him instead. So if you look, remember how in Bio 430, those of you that had me, one of the ways to find the vomer was to turn it upside down and look in the back of the nasal cavity? So those two openings split by the vomer and the ethmoid are the openings between the nasal cavity and the pharynx. So on the half of this, we're missing the perpendicular plate. So we're really just seeing one side of one opening. But those posterior spaces on either side of the perpendicular plate form the internal nares. So once we pass through that space, now we're in the throat. All right, we're in the pharynx itself. leave the nasal cavity, the epithelium that lines the nasal cavity is pseudostratified columnar epithelium. Right? Now you don't have to, those of you that didn't have me last semester, I don't require the addition of ciliated because except for a couple of places in the body, it's always ciliated. This is also given the name the respiratory epithelium. Obviously we have lots of different epithelia in the respiratory tract, and it's not the same as the respiratory membrane. That was the double wall that gas exchange occurs across the alveolus to the capillary. It involves the epithelium of both of those structures. So respiratory epithelium is just a kind of a general term for the pseudostratified columnar, if you read that. However, on a lab quiz or an exam, if I ask you to identify this epithelium, I want pseudostratified columnar, okay, as a histology structure. Now we continue that as we move into the pharynx. So the pharynx is throat. We've already used that term for the pharyngeal tonsils, the nasopharyngeal tonsils, and it's divided into 3 regions. So we have, if we take the soft palate here and straighten it out, this is what happens when you cough, or say a hard C or G sound like cookie, or not giraffe, but gone. This soft palate moves up to close off that space. And so from here up would be a division from the top of the epiglottis to that, and from the top of the epiglottis to the opening, separation between esophagus and larynx would be the bottom portion of our pharynx. Those 3 different pharyngeal structures that I indicated for you. So let me draw that across here. And then that requires that I put in the tongue. And epiglottis. All right, so A is going to be nasopharynx. Now you could say pharyngeal tonsils or nasopharyngeal tonsils, but here you have to identify it as nasopharynx because we have 2 other components of the pharynx. All right. So its epithelium is a passageway for air. I mean, sometimes when you're laughing so hard, you'll cough milk or soda or something back up your nose. But normally the nasopharynx is not a passageway for food. And so it continues to have the respiratory epithelium. So this is going to be pseudo-stratified columnar epithelium. It has a couple of structures in it. One, you're already familiar with from our last unit. The adenoids. So the quicker to write than pharyngeal tonsil. And right smack dab where my letter A is, we have the opening for the internal auditory tube, the eustachian tube. All right, notice its proximity to the palate, so soft and hard palate. This is why if your ears get plugged up, stretching your jaw or swallowing, chewing gum will help to open that up. Because it starts off bony in the temporal bone and then becomes cartilaginous, and so muscles from your palate and your pharynx attach to that auditory tube and help to open it up. Then we move into B. Can I erase this over here? You guys have that? Anybody want to take pictures first?